Good afternoon, and welcome to the Brain and Behavior Research Foundation's Meet the Scientist monthly webinar series. I'm Dr. Jeff Borenstein, President and CEO of the Foundation, and your host for today's webinar. Today, Dr. Susanne Ahmari will present the search for novel treatment targets for obsessive compulsive disorder. The Brain and Behavior Research Foundation funds the most innovative ideas in neuroscience and psychiatry to better understand the causes and develop new ways to treat brain and behavior disorders. These disorders include addiction, ADHD, anxiety, autism, bipolar disorder, borderline personality disorder, depression, eating disorders, OCD, post-traumatic stress, and schizophrenia. Since 1987, the Foundation has awarded more than $394 million to fund more than 5,700 grants to scientists around the world. 100% of all donor contributions for research are invested in our grants to scientists. I'm delighted to introduce Dr. Susanne Ahmari. Dr. Ahmari is Assistant Professor of Psychiatry at the University of Pittsburgh School of Medicine. She is a member of our Scientific Council and was a Foundation Young Investigator Grantee in 2012. Today's webinar will begin with Dr. Ahmari's presentation. This will be followed by a question and answer period. To submit your questions, please use the Questions tab on the control panel on your screen. Feel free to submit your questions at any time. Following the presentation, I will ask as many as possible in the time allotted. And now, I'm pleased to introduce Dr. Ahmari. Susanne, the floor is yours.
Thank you so much, Dr. Borenstein. It's really a pleasure to be here. I hope everyone can hear me out there in webinar land. And it's just really fantastic that the Brain and Behavior Research Foundation hosts these webinars. And I'm excited for this opportunity to, first, what I'm going to do is discuss the current landscape regarding obsessive compulsive disorder, otherwise known as OCD, as well as its treatment, and then highlight some examples from work in our own lab that we hope will lead us someday to improve treatments in the future. So my research program is focused on trying to understand the molecular, cellular, and circuit abnormalities that underlie abnormal repetitive thoughts and behaviors, which I like to show this Venn diagram because it really highlights that these are very common symptoms across a variety of different neuropsychiatric disorders, ranging all the way from things that you could classify as more simple and automatic behaviors, such as repetitive muscle contraction and dystonia, or pill rolling tremors and Parkinson's disease, all the way up through more I tried to move that little thing. Okay, all the way up to more complex series of maladaptive repetitive actions, such as you might see in something like addiction. But in particular, my lab is very focused on trying to understand the neural substrates underlying the obsessions and compulsions that are really central to OCD. I do want to highlight one thing about this diagram that I've made here, which is I've got the arrow here going to complex and, in quotes, volitional behaviors, because I want to be very clear about the fact that when people are performing compulsions or other behaviors such as you might see in these other illnesses, it's not as if they want to do them. So I don't mean volitional in that sense. What I mean is, from a research perspective, the kinds of actions that are going on in the brain, the kinds of processes that are going on in the brain when people are performing things like compulsions, may be things like decision making or selection of actions that we tend to think of as more complex things that can maybe be brought under voluntary control if you have the right kinds of treatment interventions. And that's very different, for example, than a tremor, in which you can't imagine doing exposure therapy for that. So, even though I'm probably preaching to the choir for my audience here, I do want to just very quickly, since this is a public talk, highlight that it's really important to have the message that OCD is a very severe psychiatric illness. It's not a joke, and I think this kind of gets portrayed in the media in the wrong way a lot of the time. So I do want to highlight what OCD isn't. You know, it is not a joke. It is not being obsessed with the holidays. It's not, you know, being hyper organized and having everything organized, you know, by color, for example. It's not being that person who always has Purell to give out at restaurants, you know, before people are starting to eat, although of course that could be a part of somebody's OCD. And this is just from a Twitter feed of Khloe Kardashian, which I liked the response to it, because she's saying, see how I close CD my tableware. And Chrissy says, does anyone else find it mildly inappropriate to make a medical mental disorder into a cute name brand tag? And so I think it's our responsibility as clinicians, treatment providers, researchers, et cetera, to really kind of highlight that we want to get out of our parlance that phrase, I'm so OCD that I do, you know, this thing, which is actually in the realm of normal behavior. Now, so what OCD is, according to our current DSM-5 criteria, it consists of obsessions, which are recurrent, persistent, intrusive thoughts, images, or impulses, or compulsions, typically and compulsions, which are repetitive behaviors or mental acts that people tend to engage in to reduce that severe anxiety and dread that's associated with those obsessive thoughts. And, you know, these are kind of the cut and dried criteria, but that doesn't give you the real flavor of the illness. So again, I'll turn to some kind of pop culture to give you an example or two, actually. And these are wonderful cartoons that are drawn by Lily Williams. You can find them online that actually give you a real flavor of what OCD is or what it can be for particular people. So in this particular example, I must do everything 8 times in a row. If I lose count or mess up, I have to start over. And if I don't do this, these intrusive thoughts repeat over and over and over. My parents will die. And again, the cartoon does a great job of portraying the extent of distress that's associated with this thought, because the thought is, of course, an incredibly scary one. Here's another example. I'm so OCD that I think all food is poisoned. And here we have the list of the rules for determining if food is contaminated. And then, and this is an important thing I want to highlight here, regardless of if I trust someone, I do not trust there aren't drugs in all food. And then, of course, this leads to significant dysfunction. And if she does end up eating something that she believes is contaminated, she can't function. And this highlights the fact that even when the person logically knows that there is no likelihood, no real likelihood of this particular fear of coming true, this remains a problem. This remains something that the person acts on. So OCD also is quite common and severe. So if we look at the prevalence statistics, it's quite common, 1 to 3% lifetime prevalence. And the interesting thing is, it doesn't seem to matter where you are in the world or what culture you are. There's really a relatively consistent level of OCD worldwide. There are 2 peaks of onset. One's in childhood and seems to be more prevalent in males and may have more of a genetic link. And then the other is in late adolescence, early adulthood. And so what that highlights is the chronicity. In the absence of treatment, this can be a disorder which really lasts a lifetime, which again shows us that, you know, this is something that we really need to take seriously. Now, it can also be quite severe. Patients can spend many hours per day consumed by symptoms. It can interfere with education, work, and independent living, and in many cases can be very difficult to treat. Now, I want to highlight, the next thing I want to highlight about OCD is also important, I think, from a research perspective and as well as a treatment perspective. And it is that OCD symptoms can be quite heterogeneous. And people out there in the audience who are familiar with OCD are probably familiar with the fact that there are several core types of OCD known as symptom dimensions. We have contamination-related OCD, checking-related OCD, taboo or harm avoidance-related OCD, and then symmetry-related OCD. I'm also including here, even though it is no longer thought to be a part of OCD in terms of the diagnosis as well as the neurobiology, I'm putting hoarding up simply because it's something that often gets talked about in relationship to OCD, and there is a significant amount of comorbidity. And other people, if you do, you know, online searches, both in the scientific literature as well as in just the online web, There are other proposed subdivisions of OCD as well. There was actually a question that got put in before the webinar about obsessive slowness as a potential type of OCD. Tic-related OCD has some good scientific evidence behind it as being potentially different in terms both of brain abnormalities as well as treatment response. And there's, of course, a lot of thoughts about pure obsessional OCD or pure O. And I think this is a really important thing to think about because I think it has significant treatment implications. And it's very clear from a research perspective that even though there have been a lot of studies which have tried to identify these kinds of subtypes of OCD, I think it's really still only the tip of the iceberg in terms of trying to identify shared versus distinct elements among patients who are all suffering from the same disorder. Because even if we look simply at these 4 categories here, contamination, checking, harm, and symmetry, you know, we know from longitudinal studies that many patients switch back and forth between these different types of symptoms or have different types of symptoms at the same time. And so what we're really interested in is trying to identify unifying features that kind of cross these dimensions that we could potentially attack through different targeted treatments. And one, you know, just one specific example is, you know, the idea that uncertainty about the future may have an important role in many of these different things and difficulties tolerating uncertainty. And I'll get more into this a little bit later on in the talk. Now, what I next want to highlight is kind of the current state of affairs with respect to treatments for OCD, because I think it's important for us to do that before we start talking about the research directions that people are trying to take in order to develop new treatments. So, you know, pharmacotherapy for OCD is, you know, there's a relatively standard of there's a relatively clear standard of care for this. Sorry, I just wanted to make sure you can still hear me because I just got a really weird feedback on my phone, but it looks like it's okay. So what I've done here is put up a schematic diagram of the serotonin system in the brain, and this is just, the reason that I did this is because serotonin reuptake inhibitors, otherwise known as SRIs, are still the only proven monotherapy for OCD. And this is things such as the selective serotonin reuptake inhibitors, such as fluoxetine, paroxetine, et cetera, as well as what is still kind of thought of as the gold standard in OCD pharmacotherapy, which is clomipramine, a tricyclic antidepressant, which also has serotonergic efficacy. Now, SRIs can be very helpful for people. It can lead to full remission in approximately 10 to 15% of people. And a lot of people are also partial responders, depending on which study you look at, ranging from 20 to 40%. So this is a significant chunk of people, you know, somewhere between 40 to 60% who do have some response to SSRIs. But there are a few problems associated with that. Number one is that they can have significant side effects. Number two, there has been a recent study which has indicated that there's really only about 20% remission at greater than 10 years of follow-up. And so this is showing us that even though the SSRIs can kind of put a dent in symptoms and actually can be transformative for some people, new treatment options are likely necessary with respect to pharmacotherapy. Now, there are multiple augmentation strategies, and I'm not going to go into all of the potential options that are out there, but I do want to highlight a couple of ones that are relatively commonly used and that are currently generating excitement in the field. So first we have glutamatergic agents, and I've listed some references here, and specific ones that are gaining interest in particular most recently is ketamine, which is known to provide fast-acting antidepressant relief in major depressive disorder. And there's still ongoing research being done to determine how efficacious it is with respect to OCD, with one study showing that it is efficacious. That's the first one, Rodriguez et al., and second, showing that it isn't efficacious, which again highlights the likely necessity to stratify patient populations and kind of figure out what how different patient populations might be responding differently to different kinds of treatment. And other agents which are thought to attack the glutamatergic system are also out there, including things like riluzole and acetylcysteine. In addition, a relatively common augmentation strategy is to use dopaminergic blockade and what are classically called antipsychotic drugs, although we're trying to get away from that nomenclature because they're used across a broad variety of neuropsychiatric illnesses. And these kinds of medications, they tend to block a specific dopamine receptor called the D2 receptor, and the newer variety also have serotonergic efficacy as well. So, these have been very commonly used as augmentation strategies in OCD. And again, we have some evidence from studies, including this Simpson et al., that in some cases it's efficacious, in some cases it's not, but potentially when it is, it may be because people are more likely to have comorbidities with things like tics and Tourette's disorder. So that's something to consider. Now, another of the questions that came in before the webinar started was my opinion about what is my opinion about behavioral therapy-based approaches. And I can, without any question, say that my opinion is that they are excellent. So, and what I'll do is first kind of describe, to make sure we're all on the same page, the kind of principles that are associated with exposure therapy. So I've gotten this graphic of the person on the ladder because the whole idea is that the person, in collaboration with their treatment provider, they create an exposure hierarchy. I know this is extremely small and you don't need to read through it, but it's essentially an exposure hierarchy for someone with contamination-related OCD. And so you can imagine that they're creating this thing, and it has to, the particular exposures that they're imagining here will range in terms of the amount of distress that they cause, from 0% distress level all the way up to 100% stress level. So for this particular and when you're doing this kind of therapy, the idea is to get exposures that target around a 30% subjective distress level. And then as the person masters that level through procedures of learning that are still under investigation in many laboratory studies, then you basically step the hierarchy up, and then the person attacks the next level. And so an example in this case, maybe for a 30%, would be touching a trash can in a trusted therapist's office. But then the 100% would be something like touching a toilet in a public restroom and then eating later without washing hands. So there is very, very clear evidence from seminal work done by Enofoa's lab at University of Pennsylvania at exposure. They're in collaboration with many, many people, including at Columbia, that exposure therapy can be highly effective. Approximately 70% of people in this trial had significant clinically significant responses. The challenge is that it can be very difficult for patients to complete it. That also had a 30% dropout rate on that study because it's very, very hard sometimes for people to engage in this kind of therapy. And it can be very hard to find skilled treatment providers. So this is just important to consider in terms of, yes, this treatment is extremely efficacious and has actually been shown to be, at least in this particular structured trial, better than pharmacotherapy with respect to its efficacy. So it's definitely something to have in your treatment arsenal. Now, I did just mention that one of the problems and challenges is it can be hard to find skilled treatment providers. And so I've had more and more questions lately about the use of apps for OCD treatment. And there are a lot of apps out there, and I by no means, please do not take this as either endorsement or negative endorsement of any particular app. But what I do want to share with everyone out there is a particular resource that is provided by the International OCD Foundation. If you go onto their website, what you can get is access to this thing called Cyber Guide, which is actually a project of the One Mind Foundation, another organization for research and mental health awareness. And what Cyber Guide does is it actually assesses all of these apps on a level of credibility, including, essentially what is the scientific basis for this to be working, how the user experiences, what the data transparency is, and simple things like the platforms and cost. And so you can go on there and get the most up-to-date information about apps that may be useful for OCD. But I do want to highlight that it's just important to realize that these kinds of apps are still under active investigation in terms of the research that's being done as to how efficacious they are. So still the best thing to do if you have access would be to engage in treatment with an experienced provider, but I think these can be extremely useful as tools, as supplements to when you don't have access to the therapist. Okay, so now I'm going to shift into less standard treatments, but again, from the sound of some of the questions that had come in prior to the seminar, I think probably a lot of people tuning in are people who have already had experience with the more traditional treatments and have not found it to be efficacious. So when you have kind of exhausted the list of the pharmacotherapies, the psychotherapies, etc., neurosurgical treatments for OCD are definitely an option. And there's a history of this, starting with ablative neurosurgical procedures. And the idea behind these procedures, which I'll get into more in the research section of the talk, is that all of them consistently are doing kind of a similar conceptual thing, which is making precise lesions of connections between the cortex and the striatum in OCD patients. And the cortex is a region that's very important for a lot of functions, including decision making, selecting what actions you're going to do. And the striatum, in addition to other functions, is important for carrying those actions out. And there's evidence of hyperactivity and hyperconnectivity between cortex and striatum in OCD. And so when these kinds of ablation procedures are carried out, they can actually be quite effective in centers where a lot of these procedures have been done, on order of a 50 to 70% treatment response. But of course, one of the challenges associated with this is a non-reversible procedure, and of course, it's an invasive neurosurgical procedure. So definitely not on the first line of what you want to do. Now, more recently, there's been a lot of excitement about deep brain stimulation for OCD, and at this point, it can be obtained. You don't have to be in a clinical trial in order to receive it, and there's many institutions around the country who can provide it. It can be obtained through an FDA humanitarian device exemption, and there's a lot of active investigation in terms of trying to think about what are the best targets for treatment. So the classic target, which has been studied most, is the ventral capsule ventral striatum target, which I'm depicting here. Here are the two electrodes which are being implanted into the brain. And again, it depends on the study and it depends on the particular clinical site that's being examined. But in some cases, actually in many cases, you can get up between 50 and 60% efficacy of this deep brain stimulation. And just to give you a sense of what happens, again, it's a neurosurgical procedure, so it is invasive. The electrodes are implanted, and then after the implantation, essentially these electrodes are hooked to a wire which is implanted in the chest and effectively serves as kind of a pacemaker for the brain in a way. And so the patient works with the treatment providers, including the neurosurgeon, psychiatrist, and the stimulator programmer, to try and find the most efficacious leads in stimulation frequencies for their particular symptoms. Now, there's a very recent study that came out that was really interesting. It's a smaller study, and you'll see why, though, when I describe it to you. It involves 6 patients, but they were implanted in 2 different regions. So they were implanted in both this ventral capsule ventral striatum target, as well as the part of the subthalamic nucleus called the limbic subthalamic nucleus. And so each of these patients got stimulation treatment at both of those different sites. And they did a head-to-head comparison, therefore, on how effective these 2 different sites, which have been proposed to work for OCD, how effective were they in these individuals. And these individuals all had really good treatment responses. And what was interesting was that in both of the targets, they got good responses. But in the ventral capsule ventral striatal response, it seemed to have the biggest impact on mood. And then in the limbic STN responses, it seemed to have the biggest impact on the on cognitive flexibility, which they were assessing with a particular set shifting task. And so that was really interesting and exciting, because what it's suggesting is that it kind of makes sense to us as neuroscientists that if you're stimulating particular connections in the brain, but that might be linked to different specific functions, then you might be able to specifically target different subsets of people's symptoms. And so if, for example, a particular person with OCD might have more difficulty with flexible thinking, and again, this is kind of broad strokes, and we're certainly not there yet in terms of individualized treatment. But if somebody had more problems with that kind of symptom, more kinds of cognitive rigidity symptoms, then maybe the limbic STN would be a better target for them, versus if someone had really severe comorbid depression with their OCD, perhaps the ventral capsule target would be better. So these are all really interesting potential areas of investigation. And the final thing that I wanted to talk about in terms of treatments which are currently being investigated and are areas of excitement is repetitive transcranial magnetic stimulation, or TMS, because I'm confident that this is going to become much more common soon, and have already, again, been getting lots of inquiries about it from patients. So this is a non-invasive procedure. The image that I'm showing you here is one that's set up to do more deep transcranial magnetic stimulation. And I do want to highlight it is still investigational. So this is an active area of study. It's a much more broadly accepted treatment for major depressive disorder, but in OCD, we're still at the stage of trying to figure out how efficacious it is, and again, for which kinds of patients. The principle is that you have this electromagnetic field emitted, which changes the electrical currents in the underlying cortex, and that the brain activity can be stimulated or inhibited depending on the protocol used. And one thing to understand about this treatment is it intervenes, again, as I've said here, in the underlying cortex. So unlike the deep brain stimulation, where you can put your electrodes deep into the brain, this is limited by how much access you can get to the surface of the brain. And with stronger magnets that are now being used, we can get our structures a little bit deeper, so that we can target some of the more important areas in the cortex that are the whole cortex is important, but if we can target the cortical areas that are thought to be the most important for OCD, but still we are limited to cortical areas. So in terms of what are the current investigational targets that are being used most frequently, we have initial pilot study, early studies which showed that there was some efficacy for stimulation of the pre-supplementary motor area, which has been associated with motor planning and motor control. And I'll come back to when I talk a little bit about our research. In addition, there was a very recent study published, which is really exciting, where they targeted the parts of the medial prefrontal cortex and anterior cingulate cortex, which may be important for things like error monitoring, integration of thought and motivation, how to make decisions under conflict, et cetera, as well as the interaction of anxiety with learning and habit formation. And so when they performed this TMS, they actually found significant results in the OCD patients with the sham procedure compared to the actual TMS stimulation. So they had a drop in their Y-box score of 6 points compared to 3.3 points with the sham treatment people. And this was maintained at the 1-month follow-up. So there were these significant differences between the groups, and it's important to think about this from the perspective of, okay, well, they had a drop in the assessment scale, this Y-box score. How much does that translate into the actual efficacy for the patients in their daily lives? But they also looked at essentially an assessment of overall function, and they saw that it led to improvements there too. So this is a really exciting study. Of course, it needs more follow-up as well. in terms of seeing exactly how that stimulation protocol impacted brain function. So, and I do want to make one plug for my colleague here at the University of Pittsburgh, Dr. Rebecca Price. There were a couple of studies looking at TMS in the orbital frontal cortex, one of which was positive, one of which is negative in the past. And Dr. Price is investigating the potential efficacy of this treatment and seeing if in combination with a task which helps with overriding habits, if stimulation of the orbital frontal cortex, either potentiating it or depotentiating it, may be helpful for OCD. So she is currently enrolling. So if you're interested, you can contact me directly or her, or look us up on the website for University of Pittsburgh Psychiatry. Okay, so now in the second half of the talk, I want to shift a little because, so what I was talking about was the current state of affairs, the current landscape of, what we think OCD is and how are we currently treating it. But what my lab is primarily trying to do is engage in translational research, which we hope ultimately is going to pave the way for new treatments. So how do we go about translating between humans and animal models? So there are many levels at which we can do this by taking human findings related to circuits, molecules, genes that have been found to be different in OCD, and then using this information to dissect mechanisms in animal models by looking at behaviors that are relevant to OCD in people. So in this talk today, I'm going to highlight just a few examples of the approaches my lab has used to address this challenge. And then I'll end by talking about opportunities for engagement and research to help develop new treatment approaches. So what I first want to do is use a clinical example to actually highlight the framework that we try and use in the lab. And I'll use an example from contamination-related OCD because most people are familiar with that. So if someone with contamination-related OCD saw a person with a cut on their finger, that external visual stimulus would immediately lead to an obsessive thought. I might get HIV, for example. And the uncertainty about whether or not this would be the case would lead to significant distress and anxiety and what I'm labeling as inappropriate fear. Again, inappropriate because hearkening back to the cartoon I showed earlier, it's that the person with the OCD often knows logically that there is no likelihood that this can happen. But despite the fact, at least in one model of how the illness works, this leads to significant distress and anxiety that drives rituals to avoid or decrease that anxiety. And those rituals, of course, are called compulsions. And so in this specific example, that would be repetitive hand washing to the point of developing lesions in this particular patient. Now, the really frustrating thing for patients and providers in terms of how this illness works is that these compulsions can, in many cases, actually serve their purpose, at least in the short term, of leading to temporary relief from distress and anxiety. So in the short term, the patient might feel better, but this is extremely fleeting for most people. And again, you have the obsessions creeping up again, the distress and anxiety creeping up again. And we actually have a hypothesis that this is due to impairments in classic negative reinforcement. And negative reinforcement is when removing something bad makes the brain want to do that thing again and again. And so in this case, the negative reinforcer is the anxiety. So getting rid of the anxiety ends up reinforcing these behaviors, even though the person consciously does not want to do them. And I really want to emphasize this. This is something that they do not want to do. So then you get this buildup of, I'm gesturing with my hands and I just realized you can't see that. So what we have is this situation in which we have these repeated obsessive compulsive loops. Now, stepping back from this, you can see that there's many different nodes this kind of circuit that could be abnormal in someone with OCD that could lead to problems and lead to some of these loops. So you could have excessive attention to these kinds of trigger stimuli. You could have giving increased drive into getting these kinds of obsessive compulsive loops. You could have decreased ability to exit these loops once you've started. And so these kinds of things allow us to start thinking as neuroscientists in the lab of, can we dissect the circuits that are underlying these different kinds of mental processes? And can we do it in tasks that are similar enough to those that are done in people in mice so that we can get some relevant clinical information? So, and I can conceptualize this as a problem with what I call behavior transitions, of getting stuck in these loops. And here's a quote from someone with OCD. It's like a broken machine. Thoughts go in your head, get stuck, and keep going around and around. So we want to understand that. And we think that we can use animal models to start to try to do so. So I'm just going to give a few quick examples of research in the lab, which are highlighting different approaches to attacking this and similar problems. So the first one is a circuits-based example. So we're fortunate in the field of OCD that we actually have a very rich human neuroimaging literature to turn to when we start to think about where to start looking for potential abnormalities that we can translate into animals. And we have many different studies over the years now which have highlighted hyperactivity in regions of the brain, including orbital frontal cortex, anterior cingulate, the caudate, which is part of the striatum, and the thalamus, both at baseline and with symptom provocation by looking both with PET scans and fMRI studies. And we also have evidence that when you do effective treatment interventions, when treatment interventions do work, such as SSRIs or SRIs or deep brain stimulation, that this hyperactivity tends to get toned down in hyperconnectivity. But, of course, in humans, we can't test cause and effect. So in order to start to try to tackle this question in mice, we tried to take some of these findings, and specifically hyperactivity in orbital frontal cortex and striatum, and try and translate them into an animal to test whether hyperstimulating connections between the OFC and the striatum would actually lead to abnormal repetitive behaviors. And to do so, we took advantage of what at that time was a new technology called optogenetics, and it takes advantage of light-activated ion channels that are isolated from bacteria. And if you put those, they basically respond to light by opening up, letting sodium flow in, potassium flow out. And neuroscientists realize that if you put enough of those channels into selective populations of neurons, as I'm schematizing here, then effectively, if you can implant a light probe into the brain, you can shine light on this and then essentially turn neurons on and off by turning the light on and off. And so armed with this system, you can basically, in a freely moving animal, and mice in particular in the studies we were doing, you can activate neurons or inhibit them and then look at the impact on behavior as they're engaging in normal behavior. So we injected one of these opsin molecules into the orbital frontal cortex, put our light probe into striatum, and then this is a diagram of being able to illuminate these projections so that we're effectively trying to create that hyperactivity that we would see in people. And this is just showing electrical traces showing that with pulses of blue light, you do get stimulation in the brain. And so then we had to figure out, okay, when we do that stimulation, do we actually see anything that's relevant to OCD in people? And that's a big question, and I don't have time for another webinar, so I'll have to go through it relatively quickly. But of course, these are mice, and we can't interrogate them with words, so we have to do the best that we can by interrogating them with specific behavioral tests. And so, as I said, I don't have time to get into all the details, but suffice it to say we have relatively good evidence that perseverative grooming behavior in mice may have relevance to OCD in people, as may perseverative locomotion behavior. Difficulties with filtering stimuli out of the environment has been shown in work that I did in collaboration with my mentor Blair Simpson, and others have also shown that there are deficits in this process in people with OCD. And then there are also impairments in flexible learning. And this is showing you a task that'll come up a little bit later as well. It's called reversal learning. Basically, the mouse has to learn to press one lever in order to get a delicious chocolate pellet. But then we're mean and we switch the rule on them. And then they have to learn to press the other lever in order to get the chocolate pellet. And in similar but more complicated tasks in people with OCD, people tend to have either behavioral abnormalities or abnormal activation patterns in the brain when they're engaged in this kind of task. And OCD often is associated also with significant anxiety. And so we have ways of measuring anxiety-related behaviors in animals by seeing how much they venture out into more perilous areas of arenas like the center of the open field or the arms of an elevated plus maze. So armed with all of these techniques, we basically hyperstimulated that projection, and we saw if we repeatedly stimulated over the course of multiple days in a row, that we saw something very interesting, which is on the first day, we really didn't see any difference between the stimulated and unstimulated mice, but over the course of multiple days of stimulation, we saw an increase in grooming behavior, which again, we think does have some relevance to the illness. Now, what's also interesting to note is that these changes that we saw, they actually weren't happening right during the stimulation, they were happening afterwards. And so this really suggested that maybe we were changing some kind of plasticity process in the brain, which we verified by doing recordings at the same time in the brain and showing that over the course of multiple days, for each amount of electricity you kind of pulsed into the brain, you would get more out. And not only that, if we came back 2 weeks later, we still saw a persistent increase in the amount of grooming that the animals were doing in the stimulated animals, even though they'd had no intervening stimulation, although it was significantly decayed over time. So what this was suggesting was that brief but repeated pulses in these circuits that we thought were relevant to OCD did lead to the progressive development of an abnormal repetitive behavior. Of course, we still have a lot of questions about mechanisms, and we're interested in whether this information can tell us anything that's relevant to treatment. And so, specifically, what I wanted to highlight is kind of conceptually, how this could potentially be, this information could potentially be used in that context. So, if you think back to the neurostimulation-based studies I was telling you about earlier, you remember the point, the strongest evidence that we have, at least currently, is for deep brain stimulation, but the problem, of course, is that particular treatment is invasive. It's targeting these deeper structures of the brain. And even though neurosurgical procedures are quite safe these days, it's still a really high bar to meet criteria for that treatment. But I've also talked to you, about these circuits that are kind of looping between the cortex, striatum, thalamus, and back again. So if there's some activity pattern in the striatum we want to change, maybe we could do that at a distance. That is by using something like TMS in the cortex to stimulate particular patterns of activity at the surface of the brain that are then reflected down amongst these other circuits. So we can use the system that I described to you to test this kind of idea, and I'll just tell you about new technologies that we can use to do so. So here's one example of a recording device where we can record in the orbital frontal cortex and the striatum at the same time as we're stimulating, so we can get a sense of how the communication between these two different structures is changing as the stimulation evolves. And also in other work that's ongoing in the lab, we're using a relatively new set of tools, which are these miniature microscopes, where you can implant lenses deep into the brain, and then the mouse can wear this tiny microscope as it's running around engaging in normal behavior, or abnormal behavior, actually. And this is me holding one in my hands, and you can see it's quite tiny. It weighs only 1.9 grams, so it's quite easy for an animal to carry around. And I'll just show you an example of one of the videos that you can get from this. This is a video from a mouse that's engaged in compulsive grooming behavior. And what you will hopefully see, although it's looking, there we go, is that with each of these flashes is corresponding to one neuron which is being activated. So you can see this communication of neurons in real time as the animal is engaged in a compulsive behavior. So we can now follow this activity and see what the relationship is between the activity and the abnormal behavior. And this may give us clues about how the different cell populations are working together to generate abnormal behavior, and therefore it might give us clues as to what kind of stimulation we need to do to disrupt these abnormal patterns to kind of get out of the compulsive rut. And this could potentially be informative for directing stimulation paradigms for other kinds of for the kinds of TMS or DBS treatments that I was talking about. And importantly, this kind of technology allows us to track individual neurons over time so that we can see how their activity is changing in response either to the development of an abnormal behavior or in response to treatment of an abnormal behavior. So we can get insight through this into how particular treatments might work as well. So next I'm going to move on to OCD molecular changes. Now, and this is kind of in the molecules and genes purview. Now, it's very clear from twin and family studies that there is a significant genetic component to the etiology of OCD, with heritability of OCD being on the order of 40 to 60%, and it being, as I mentioned earlier, higher in children than adults. And typically, genome-wide association studies are being used these days to identify common genetic risk factors for illnesses. Now, there have been genome-wide association studies done in OCD, and the International OCD Foundation Genetics Collaborative published some of these results in 2018 from 2,688 cases, 7,037 controls. And unfortunately, in this case, there were 0 statistically significant risk genes. Now, you may be thinking, well, then where's your evidence that there is a genetic component of OCD? But I want to highlight something here that will help probably explain this. So if we take the same kind of example for schizophrenia, which was published in 2014, their study used on the order of 37,000 cases and 113,000 controls, significantly more power to detect significant risk genes. And there they got 108 significant risk genes. So, you know, we're dramatically underpowered in our studies here, which is hampering our ability to find these genes. And in part, that's because I think more investment needs to be put into OCD research. Now, in the meantime, there's also been good progress in ongoing studies that are attempting to find rare OCD genes that have large effect size, that, you know, maybe even though these rare mutations may only be affecting small percentages of people, of the larger population of people who have OCD, they can often give us a lot of insight when we translate them into animal models. And so this is a really exciting a bit of progress. In the meantime, though, because we do have this real gap in terms of what we understand about the genetic and molecular processes involved in OCD, we decided to take advantage of the brain tissue donation program at University of Pittsburgh to see if we could find people with OCD who had donated their brains after their death, and then look at gene expression in regions of the brain thought to be important in OCD, including OFC, caudate, and nucleus accumbens. And where we decided to start focusing was at the synapse, and specifically at excitatory synapses, because despite the fact that there weren't significant hits in the genome-wide association study, there was evidence that some of the molecules in the excitatory synapse were like the closest to being significant hits. And there's also been evidence from pharmacologic studies and lots of theories that these excitatory synapses are important. So this was work spearheaded by Jean Piantadosi and Brittany Chamberlain, two wonderful graduate student and undergraduate students in my lab. They combed the brain bank. They only found 8 subjects who donated their brains with OCD, and 8 comparison subjects were then included. Here's the statistics on them. You can see they're very well matched in terms of age, sex, post-mortem interval, and brain pH, other things that impact the quality of the tissue. A majority of the patients were on antidepressants at time of death, so that's something to keep in mind. And also there was significant comorbidity, as expected, with things like major depressive disorder, OCPD, and anxiety disorders. So we honestly didn't go into this study thinking that with this low of a number of subjects we were going to see really strong effects, but we have to start somewhere. And so we took these regions of the brain, medial and lateral OFC, caudate and accumbens, sliced fresh frozen tissue, extracted RNA, and then performed qPCR in order to look at the abnormal molecules involved. And this is a busy slide, but what I want to focus on is that in the black are the controls, in the red are the people with OCD, and you can see that if you look on this side, these are the orbital frontal cortex panels, and then this side is the striatum. You can see, compared to the unaffected comparison subjects, the OCD subjects had lower levels of many of these excitatory synaptic proteins across the board in the OFC. And in the striatum, there's evidence for that, but it's not as strong. And if we look at some other transcripts, which are important for signaling in these synapses, these are receptors and transporters, you see the same pattern. In the OFC, you have this downregulation. And then in the striatum, there is some evidence for downregulation, but it's not as strong. And we had some hypotheses about inhibitory synapse transcripts potentially be involved, in part because of prior work that's been done in Tourette's. But in fact, when we looked at that, there were not that significant of differences. So when we look at this all told, what we see is if we pool all of the synaptic structure transcripts together, we see a significant decrease in their expression in medial and lateral OFC compared to the caudate and the nucleus accumbens, and the same thing in the receptor and transporter composite score. So this was really exciting to us because what it's suggesting is that the OFC, which we'd always thought was important in OCD, might be an interesting molecular hub to look at. But surprisingly, the directionality of these transcript changes was actually going in the opposite direction of what I at least was initially predicting, because so many of the studies have demonstrated hyperactivity. So what this is suggesting as an intriguing but completely speculative possibility is that there could be upstream thalamic pathology in the OCD subjects, and this is kind of a schematic diagram of what that might look like, with the hyperactive thalamus kind of driving the OFC and causing the OFC to try and turn down the volume on that increased input from the thalamus. And of course, there is evidence that the thalamus is hyperactive in OCD. So this is something we're actively investigating with support from the International OCD Foundation and the One Mind Foundation. Now, finally, in just the few remaining minutes, because I do want to leave some time for questions, I want to highlight how transgenic models of compulsive behaviors can be of utility in uncovering mechanisms that may be important for treatment targeting as well. And so now we're on the level of circuits and genes. So what I'm going to show you now is work that we've been doing recently, taking advantage of a particular transgenic model, which is called the SAPAP3 knockout mouse. So we're knocking out a particular protein, a particular gene that is helping the synapse, this excitatory synapse, stay together. And the knockouts were actually made by Guoping Feng's group, and the references are here. And when you knock that gene out, it leads to compulsive grooming behavior that persists despite negative consequences to the mouse. One thing I do want to note, and again, I want to be very clear, it's not that we think SAPAP3 is the quote-unquote OCD gene or anything of that sort. This is a useful model system to dissect circuits and to dissect mechanisms. But what is interesting is when we do look in our post-mortem samples, we see consistent downregulation of this gene across all the different regions that we looked at. So there's at least some parallel and some potential relevance there. We know that if this gene is rescued in the striatum, it actually reverses the phenotype, and we also know that floxetine reverses the phenotype. So this is a good system to start to explore what's actually happening to lead to the striatal hyperactivity that is seen in these mice. And so one of my graduate students, Victoria Corbett, investigated this using ex vivo electrophysiology-based approaches, because she was really interested to start to try and figure out what might be driving the abnormal activity in the striatum as well as the abnormal behaviors in these animals. So she injected a tracer that goes backwards into the striatum and then saw, okay, what are the regions that project to this region of the striatum? And she saw, surprisingly, that in addition to the OFC, which we were expecting was going to go there, she also saw projections from this area called M2 in the mouse, which is equivalent to one of the areas in the human that is currently being used for transcranial magnetic stimulation targeting, the pre-SMA. And you can just see the quantification of these here. Now, what she also found, then wanted to look at that M2 region more closely, is that when she pulsed projections from that region to the striatum in normal mice versus these knockout mice, you see immediately there's a much larger pulse coming out in the knockout mice. And you can see this quantified here. These projections are much stronger and they're much more reliable, so more likely to be activated. And it looks like this is happening at the level of the post-synapse. And so that changes are happening in that postsynaptic area in the striatum. So this was intriguing to us because it did suggest this idea that these strengthened projections from this supplementary motor area to the striatum could potentially be important for pathology and therefore potentially important for treatment. Now, very quickly, I've been talking a lot about patient stratification and heterogeneity and how that might impact treatment response. We can also attack this in these mice. So Lizzie Manning, one of my postdocs, she looked at this reversal learning task that I described in these knockout mice. And I'll speed through that part because I already talked about it. But when you look at the wild type mice compared to the knockouts, what you can see is they're able to learn that first rule, i.e. that I press a lever and then I get a chocolate reward. They learn that equivalently. But when you switch the rule on them, the knockouts have a lot of trouble with that rule. And then if you look at that more closely and separate out the populations of animals, you can see that some of those knockouts perform almost identically to those wild type animals, and some of them completely fail. And we had between 40 and 50% failure rate. And so now this is really interesting, right? So these animals, which are genetically identical, some of them fail, some of them don't. It's not related to how much they groom. It's not related to how well they learned that new rule. But still, there's this striking but variable deficit in reversal learning. And so now Lizzie is using those miniature microscopes, which I described before, to track what's happening during reversal learning in an area of the brain and the prefrontal cortex that's thought to be important for this. And basically, again, this is ongoing work, so I'm just going to show you an example of the kind of things that we can get from that. We can basically image the animals when they're engaged in the compulsive behavior and image them when they're engaged in that perseverative reversal learning behavior and try and get an idea of the mechanisms that might be underlying these different things. So these are the kinds of strategies that we're trying to use. And of course, the overall goal is to develop improved neuroscience-based treatments for OCD. You know, can we take information from our stimulation findings in animals and someday translate that into helping those neurosurgeons and psychiatrists who are trying to develop new stimulation protocols for people? Can we use the microscopy work that I've told you about, in which actually this is stuff I haven't told you about today because again, it's still ongoing, but you know, looking at fluoxetine treatment response and seeing what happens in cells over time so that we can predict potentially how different, whether a responder versus a non-responder is going to react in a different way within their striatum. So I want to end by really emphasizing what I see as the benefits of including people with lived experience, people who are living with OCD in research. You know, it's really important, I think, for us as clinicians and researchers to try and engage the people who have the illness. And a very concrete example of this is in the study that we're just recently recruiting for. As of this week, we just got our first OCD participant scanned. And this is an ongoing study in which we're essentially testing that idea, and this is with me and Dr. Rebecca Price, as well as other researchers here at the University of Pittsburgh. We're testing to see whether or not that hypothesis about negative reinforcement being important for OCD is bearing any fruit, and we're doing this using a brain imaging study. So we're actively recruiting. I do want to make that plug, but the reason I bring this up now is that this study was initially inspired by interactions I had with a patient in my practice. We were talking about kind of what the motivation for doing the compulsive behavior is like. So I think the more we can have these kinds of conversations with patients, tell them what we as researchers are thinking about, and then they can tell us what they're experiencing as patients and kind of feed into our research ideas, I think it's going to be beneficial for everyone. Now, if you do want to consider participating in studies, you know, you can always reach out to your you know, whatever research organizations exist in your area. But even if you don't live in an area with a research organization, university, et cetera, the International OCD Foundation has resources available listing lots of studies, including online studies that you can participate in. And I want to end by encouraging everyone to really please consider brain donation. It's exclusive. That and organ donation do not go hand in hand. So if you've checked your organ donor card, that does not include brain donation. So please consider going to the braindonorproject.org. Without this kind of information, we wouldn't, or without this kind of resource, we wouldn't be able to get this information about potential molecules involved in OCD that now is very exciting and might lead us towards new targets. So with that, I'll end by first thanking the patients and their families for the generous gift of brain donation, as well as my lab members who are amazing and couldn't do any of this work without them, and the funding sources, of course, highlighting the Brain and Behavior Research Foundation. So thank you very much for your attention.
Well, let me say, Susanne, that was an excellent presentation. I appreciate you taking complicated issues and making them understandable to the general public. So thank you for that. Thank you for the research that you've been doing.
Thank you.
I want to ask you about an important issue that a number of people asked about, which is the issue of early diagnosis, and that many people suffer in silence with OCD symptoms without seeking help. I'd like you to talk a little bit about that.
Yeah, no, it's a really huge problem, and I think I think the most recent stats on that are something like the average person will wait 9 years before seeking help. And again, I'm not sure exactly how accurate that is, and it's honestly a hard thing to measure. But and this is a big problem with OCD. I would say with mental illness in general, but I think OCD in particular, because for many people, the obsessions, the obsessive thoughts in particular, as well as the compulsive behaviors, but they come along with a lot of shame. Because these are intrusive thoughts that people do not want in their heads, and they're often incongruent with anything that's in their normal level of experience. And I'll give a couple of examples because I think it really helps to normalize these things. These can be thoughts about wanting to harm family members, for example, wanting to harm your newborn baby, even though you truly have no intention of doing so, and no desire to do so. It can be intrusive thoughts about sexual urges that seem taboo. And I mention these two specifically because these are the ones that people are often the most fearful about coming to treatment providers for help with, because they worry that they're going to be judged, or they worry that they're, even that, if they admit that they have intrusive thoughts about pedophilia, for example, that they'll be locked up, right? So it's important to recognize that these are symptoms of an illness and that they can be treated, and that there is hope for treatment. So I think the first part is recognition that this isn't the person's fault. It isn't a moral issue. It isn't anything like that. That everyone actually does have random intrusive thoughts that pop into your head. But for people with OCD, either they're more frequent, more intense, or they're more difficult to get rid of, or people attach more significance to them. So I think the more we can kind of take the judgment out of the thoughts and kind of put that to the side and really just focus on the fact that this is a medical condition. And if you are suffering, you can seek treatment and look for people who can help you to gain tools and gain access to treatments to start to diminish those thoughts and compulsions.
I think that's very good advice. Let me ask you one additional question before we finish up. Are there things that a parent can look for in their child to show that, because you discussed the two sort of hubs of when it may start, what behaviors would a parent see in their child that may be a cause for concern?
Yeah, it's an excellent question. I do want to preface this by saying I'm not a child psychiatrist, but I still think I can offer some advice here. So, and it gets tricky, right? Because children in general, like as a rule, there are normal developmental stages where they're engaged in what you could even classify as, definitely repetitive behaviors, and whether or not it's compulsive is another story, right? But ordering, ritualizing, needing to do the same things over and over again, needing to be read the same story 20 times in a row, these are normal parts of childhood development. So paying attention, though, to kind of what's normal for your child and what's normal for your child's peers, and also to levels of distress and anxiety that are happening in the child, I think is a really important thing to note. Because again, in kids, if you're having lots of ritualized behaviors and a lot of distress and anxiety, then I think it's not a question that you should want to at least get it checked out. It doesn't hurt to get it checked out. And so in general, that's what I would kind of say in terms of just as a broad general rule. I think, we need to think about engaging mental health care in our country in a more, in a more standard way, as opposed to kind of in a last minute, when the symptoms are fully evolved way. So if you have concerns about your child in any way, it's always good to just talk to your pediatrician about that. But specific things, of concern when you should definitely get something checked out is if it's acute onset in any way. If you're noticing that it's associated with motor tics, abnormal motor behaviors, etc., then that's something to definitely think about. But, again, in kids as well, they will often hide these symptoms and, know that they don't seem quite right. So the more you can kind of get your child to open up about what they're doing and why they're doing it, which can be very hard to do, but the better off you may be.
Also very good advice. I want to again say thank you for all the work that you've done and continue to do. It certainly offers tremendous hope for new treatment so that more people are able to benefit from treatment. I want to also thank everybody who joined us today. All of the research we fund is made possible through private donations, so please consider making a contribution by visiting our website, bbrfoundation.org, or calling us at 1-800-829-8289. This webinar has been recorded. If you've missed any portion of the presentation or would like to share it with family or friends, please visit the Events and Webinars page on our website. I hope you'll join us again next month when Dr. Lynette Averill, Associate Research Scientist in Psychiatry at the Yale University School of Medicine and Research Psychologist at the National Center for PTSD, will present Searching for Biomarkers of Stress-Related Mental Illness and Suicidality. This webinar will take place on Tuesday, July 9th at 2 p.m. Eastern Time. Once again, thank you for joining us and enjoy the rest of the day. Take care.